Fine. We are Mike? Yes. We're back. Students, can you hear me now? Any response? Okay. So we are discussing about the last part of the respiratory, the system, that is the disorders. Yes, good. We're talking about the bacterial and the viral diseases. Good. Almost like we're nearing at the closure of the end part of this chapter. Within a couple of minutes, we're going to complete this chapter as well. All right. So we're talking about TB, the bacterial disease. The causative agent is Mycobacterium tuberculosis. Pneumonia, which caused by Streptococcus pneumoniae. So you how can differentiate is either they ask the causative agent and the disease, or they'll ask the symptoms actually. So in the textbook Human Health and Disease, a particular chapter, you would have seen like the symptoms they would have given, right? Where you could see in the sputum actually. If you take the sputum of the patient of the TB patient, the sputum, if you take, can you tell me like what type of stain that usually you perform to confirm the presence of the mycobacterium under the microscope? Can someone tell me what is the name of the staining technique? Staining technique that usually they perform with the sputum sample of the TB patients. What is the name of the staining? Can someone tell me? Yes. So, what is the name of the staining technique? So, usually for bacteria, you perform gram staining technique, but for mycobacterium, it requires some sort of like the differential staining technique is required. The differential staining. So, what is the name of the staining that you can say? So that is for the Mycobacterium tuberculosis. What is the sample that you collect for the TB? Is a sputum sample. It's a regurgitated, repeated, like cough. Then definitely the doctors prescribe for recommend the sputum sample. With that sputum sample, they perform a staining, and with that staining under the microscopic examination, if they confirm the Mycobacterium this bacilli, then the patient is confirmed to be TB. Then the doctors prescribe or continue with the treatment, right? So Streptococcus pneumoniae, usually doctors can take the X-rays. Like X-rays itself, we can differentiate. As far as TB patients, X-rays you take, like the lung part will be entirely cloudy, right? With X-ray itself, like experienced doctors can differentiate whether it is pneumonia or it's a TB. TB, the entire lung part, the X-ray part seems to be cloudy, whereas in case of pneumonia, where like string kind of, the X-ray itself shows a kind of string line kind of arrangements that you could see. With that itself, with the X-ray itself, preliminarily we can identify whether the patient has got affected with TB or pneumonia. And we come to the virus actually. Most of the viral groups which cause the respiratory infections. But thing is, we have upper respiratory tract infections, lower respiratory tract infections. So we call it as URT and LRT, upper respiratory and lower. Few organisms have the ability to cause only the upper respiratory tract, and some which go even deeper inside and will creep inside the alveoli, and this can even cause like lower respiratory tract infection. Recently we talk about like this corona, even the corona which cause both upper as well as the lower respiratory tract infections. This can even reach till alveolar sac, which block the sacs, and it prevents the exchange of gases. Due to that only, like the experience, I mean the infected individuals have suffocation and breathing difficulties. So it means that understood, right? So this particular corona will have the ability to cause both upper and the lower respiratory tract infections. But many of the viral groups are the bacterial, which cause either upper respiratory tract or lower respiratory tract infections. So by this, we're coming to the end. We're completing this particular chapter, that is respiration and breathing exchange of gases, right? So then after this, like we're going to start with the next chapter, that is locomotion and movement. So far what are the things that we discussed is We start at the beginning of the respiration takes place, inspiration and expiration. You ask me what is the name of the staining. So the name of the staining is acid fast staining. Acid fast staining. Acid fast. Acid fast staining technique. But normal technique is actually um, normal is gram staining, because why the gram staining is a differentiate gram positive and gram negative. Gram staining will not work out for Mycobacterium tuberculosis. It requires a separate special staining technique that is called as acid fast staining, because we are using a strong acid here, and also we're using a dye in acid fast staining is carbon fusion. The name of the dye we are using here is a carbon fusion, and we heat fix the smear. We heat fix the smear, and then we'll see under the microscope is gram, negative, the pink color bacilli that we can see. By that we also have the count actually, like the range of the bacilli. Then we confirm whether the patient is affected with the tuberculosis, whether he or she is in the acute stage or it is in the chronic stage. Sputum, what is a sputum, right? Sputum is like the exaggerated like thing which is coming out of the upper respiratory tract is what we call it as a sputum. The most common thing. You can ask me like if the sputum is not coming right, then what is actually like the choice of the sample collection? The person is like repeatedly the patient is coughing, but the patient is telling like the sputum is not coming. Then what is actually the choice? As a technician, as a microbiologist, what do you prefer apart from the sputum is not coming? Then what's a choice of collecting the sample? Can someone tell me? The sample is actually we take the throat swab. So we have the swab, we insert the swab inside the mouth, and we collect the swab, and from that we culture actually. So we culture in not in the normal medium. We have the specialized medium for cultivation of Mycobacterium tuberculosis, that is LJ medium. So the medium for the cultivation of the Mycobacterium is LJ medium. So this is for cultivation. So cultivation of Mycobacterium tuberculosis. LJ stands for Lowenstein-Jensen medium. Lowenstein-Jensen medium. So this medium is a special medium for the cultivation of this Mycobacterium tuberculosis. This will not grow in a normal medium like other bacteria like mutant agar, Sarcoidosis agar, like a fungal species. This will not grow in a normal medium, and also this will not grow in like just 2 or 3 days. It takes like at least a minimum of like 7 to 10 days of time. So it grows even like more than it take more than even that period of time. So that is what actually about the Mycobacterium tuberculosis. But Mycobacterium leprae, that is a Hansen disease that we call. We don't get confused with Mycobacterium leprae and Mycobacterium tuberculosis, because both comes under the same genus. But Mycobacterium tuberculosis causes TB. Mycobacterium leprae, which causes leprosy. Mycobacterium leprae has no connection with the respiratory diseases. Okay, so upper respiratory tract, you ask for URT and LRT, that is upper respiratory tract and lower respiratory tract. You ask LJ. I mentioned LJ stands for Lowenstein-Jensen medium. So Lowenstein-Jensen medium is the special for, the medium for, because each and every bacteria, like, for example, you have studied about blood agar. For example, you go for a streptococcus and all, like, Streptococcus is actually like, you can ask me which medium I can go with. We go with the special medium, like, you go with the enrichment medium. You go with the blood agar and all, like, which requires for the Streptococcus, right? How the Streptococcus look like? Streptococcus, when you take the swab and cultivate in this particular medium in the microscope, you could see the Streptococcus as a gram positive cocci. This is actually, you would have studied, this would have seen the gram positive cocci in chains actually. It's in a chain. But this Mycobacterium, like, it's like a basil, like the pink color basil you could see after the staining, like acid fast staining. By that also you can differentiate whether it's a streptococci or it's a acid fast staining. But the acid fast staining, you could see the basil like this. But in case of streptococci, how you can identify the streptococci. The streptococci will be in the form of a chains actually. The cocci, it's actually purple color, positive. It's a gram positive cocci that you can do. For this, you can do the normal staining. Streptococcus, you can do the gram positive technique itself. By that, you can identify the chain-like structure that confirms it's a streptococcal infection. Fine. So with this, we are coming or winding up this particular chapter. That is respiration and that is exchange of gases. Now we move on to the next chapter, and possibly by today you'll be getting the test papers on this particular chapter. If anything that we didn't cover in this chapter, like if you have any doubts and any questions, please feel free and don't hesitate to ask questions unto me. So I'm happy to clarify those doubts as far as this chapter is concerned. Now we move on to the next chapter, that is locomotion and movement. The next chapter is locomotion and movement. This is the next chapter, that is on human physiology. Locomotion and movement here we categorize into two parts. One is your muscular system, other one is the skeletal system. So possibly today we try to complete first the muscular system. Maybe tomorrow we'll start with the skeletal system. As far as the locomotion and movement is concerned, so we're going to discuss like the first is actually locomotion and movement. So what is the difference between locomotion and uh, uh, movement actually? Okay, so you ask me sputum is a method of staining. No, no, sputum is a ஸ்பூட்டம்ன்றது என்னன்னு கேட்டீங்கன்னா நம்ம வாயில இருந்து வர சளியை தான் நம்ம என்ன சொல்லுவோம் கேட்டீங்கன்னா ஸ்பூட்டம் சொல்லுவோம். ஸ்பூட்டம்ன்றது ஸ்டெயினிங் கிடையாது. ஸ்பூட்டம் வந்து சளியை தான் நம்ம என்ன சொல்றோம்னா ஸ்பூட்டம் சொல்லுவோம். சம்டைம்ஸ் நீங்க இந்த பாராசைட்டாலஜில பாத்தீங்கன்னா ஸ்டூல்னு சொல்லுவாங்க. ஸ்டூல்ன்றது வந்து ஃபீசஸ் அதான் என்ன சொல்லுவாங்கன்னா ஸ்டூல் சாம்பிள் சொல்லுவாங்க. இந்த மெடிக்கல் டேர்ம்ஸ்ல வந்து ஸ்டூல் அப்படின்னா ஐயோ ஸ்டூல் என்னன்னு தெரியல, யூ ஷுட் நாட் பிளிங்க். ஸ்டூல் இஸ் நதிங் பட் த ஃபீக்கல் சாம்பிள். அதே மாதிரி ஃபார் எக்ஸாம்பிள் லைக் ஸ்பூட்டம் அப்படின்றது வந்து சளியை தான் நம்ம என்ன சொல்றோம்னா ஸ்பூட்டம். அதுல வந்து மியூக்கஸ் அலாங் வித் த பாக்டீரியல் ஸ்பீஷிஸ். அதை ஸ்டெயின் பண்ற டெக்னிக்கை தான் நான் என்ன சொன்னேன்னா ஆசிட் ஃபாஸ்ட் ஸ்டெயினிங். இது எதுக்கு மட்டும் கேட்டீங்கன்னா மைக்ரோபாக்டீரியம் ஆசிட் ஃபாஸ்ட். அதனாலதான் மைக்ரோபாக்டீரியம் என்ன பேசிலைன்னு சொல்லுவாங்கன்னா ஏஎஃப்பின்னு சொல்லுவோம். ஏஎஃப்பின்னா ஆசிட் ஃபாஸ்ட் பேசிலை. அது இன்னொரு பேர் இருக்கு, ஆசிட் ஃபாஸ்ட் பேசிலை இஸ் அதர் த காமன் நேம் ஃபார், பிகாஸ் இட் டேக்ஸ் ஆசிட் தட் இஸ் a கார்பன் ஃபியூஷன். அந்த ஸ்டெயினை எடுத்துக்கிறனாலையும் ஆசிடையும் எடுத்துக்கிறனாலையும் அதை ஆசிட் ஃபாஸ்ட் பேசிலை அப்படின்னு நம்ம சொல்றோம். சரிங்களா? ஸோ ஸ்பூட்டம் இஸ் நாட் ஆக்சுவலி தி ஸ்டெயினிங் டெக்னிக். ஸ்பூட்டம்ன்றது சரி. Next we move on to the locomotion and movement. Okay, so as far as locomotion க்கு movement என்ன difference அப்படின்னு கேட்டீங்கன்னா, so what exactly is the movement means? Movement is actually the change in the posture of your body parts. உங்க பாடி எல்லாம் நின்னுட்டே for example standing here and just moving and rotating moving my head, I'm talking and I know my visceral organs like is changing its posture and performing its functions. All these are actually we call it as a movement. But locomotion is actually something when the living organism switch over or literally move from one place to another. Literally ஒரு இடத்துல இருந்து இன்னொரு இடத்துக்கு மூவ் ஆச்சுன்னா then that we call it as a locomotion. So you need not confuse what is a locomotion. ரெண்டுமே சேம் கிடையாது. Locomotion and movement are different. Movement is actually within the body parts. How actually the organism, the posture, like the movement is taking place. Actually, how the visceral organs it's moving, everything it comes under the movement without like literally switching over from one place to another. But locomotion where the living organisms literally move from one place to another. So in this chapter we're going to discuss about this type of movements only. Okay, sputum and mucus is the same. இல்ல ஸ்பூட்டமும் ஸ்பூட்டம்ல இருக்கிற ஒரு சப்ஸ்டன்ஸ் தான் உங்க மியூக்கஸ். உங்க the larynx the pharynx regionல இருக்கிற வர செக்ரீஷன்ஸ் இருக்கு பாருங்க, அதுல இருந்து வர செக்ரீஷன்ஸ் பேர் தான் மியூக்கஸ். அந்த பிசு பிசுப்பா ஒரு தன்மை வருது பாத்தீங்களா? ஈவன் உங்க சலைவோட எடுத்துக்கிட்டீங்கன்னா சலைவோட கன்சிஸ்டன்சி பாத்தீங்கன்னா கொஞ்சம் ஸ்டிக்கியா இருக்கு. இந்த கன்சிஸ்டன்ஸ் ஸ்டிக்கியா இருக்கிறது காரணமா இருக்கிறது தான் என்னது? இட்ஸ் a protective membrane that the mucous membrane ல இருந்து ப்ரொடியூஸ் ஆகிற சப்ஸ்டன்ஸ் பேரை தான் மியூக்கஸ். மியூசின் என்ற சப்ஸ்டன்ஸ் மூலமா ப்ரொடியூஸ் பண்றது தான் மியூக்கஸ். மியூக்கஸ் வந்து ஸ்பூட்டம்ல இருக்கும் அவ்வளவுமே தவிர ஸ்பூட்டமும் மியூக்கஸும் டிஃபரெண்ட். But what is the pH of the sputum அப்படின்னு பாத்தீங்கன்னா இட்ஸ் யூசுவலி ஆக்சுவலி இது pH செக் பண்ண மாட்டாங்க. பட் இருந்தாலும் நீங்க pH செக் பண்ணி பாருங்க. ஏன்னா அந்த ஸ்பூட்டம்ல இருக்கிற இதுவை பொறுத்து சில நேரத்துல வந்து ஸ்பூட்டம்ல வந்து பாக்டீரியா இல்ல, சில நேரத்துல ஸ்பூட்டம்ல வந்து பிளட் டிங்சர்ஸ் இருக்கு, சில நேரத்துல வந்து வெறும் ஸ்பூட்டமா இருக்கு. அப்படின்னு பார்க்கும்போது pH வந்து வேரி ஆகும். எப்பவுமே ஸ்பூட்டம்க்கு வந்து pH வச்சு நம்ம செக் பண்ண மாட்டோம். இன்கேஸ் ஜெனரலா ஸ்பூட்டம் எடுத்துட்டீங்கன்னா அது ஆக்சுவலி வந்து 6 to 7ல தான் இருக்கும் ஸ்பூட்டம். பட் யூசுவலா pH செக் பண்ண மாட்டோம். ஏன் ரீசன் அப்படின்னு கேட்டீங்கன்னா ஸ்பூட்டம்ல வந்து சில நேரத்துல மியூக்கஸ் கொஞ்ச நேரம் கம்மியா இருக்கும். இட் மே பி லைக் மோர் பிளட் டிஞ்சர்ஸ் இருக்கலாம், மேபி பாக்டீரியா இருக்கலாம். ஸோ அதெல்லாம் ஃபிளக்சுவேட் ஆகும். pH ஃபிளக்சுவேட் ஆகிறதுனால யூசுவலா pH வந்து நம்ம ஸ்பூட்டம்க்கு செக் பண்ண மாட்டோம். சரிங்களா? ஓகே, சோ மியூக்கஸ் லொகேட்டட் இன்சைட் த ஸ்பூட்டம். நோ, ஸ்பூட்டம்ல இருக்கிற ஒரு சப்ஸ்டன்ஸ் பேர் தான் மியூக்கஸ். அவ்வளவு. So sputum la irukra substance பேர் தான் mucus. Even fetus எடுத்துக்கங்க, feces ல எடுத்தீங்கன்னா diarrhea dysentery வந்தா கூட you would have noticed in case of dysentery you could see the content of mucus. Actually it gives the stickiness to the feces, which can stick it to the containers. Like ஏன் அப்படி ஸ்டிக்கியா இருக்கு அப்படின்னு கேட்டீங்கன்னா, this is because of the mucus substance actually. So mucus is actually the secretion that you could see in the fluids, like you could see in the sputum or you could see in the fecal sample. Fine. Okay. ஸ்பூட்டம் சே தமிழ் மீனிங் சளி. சளியை தான் நம்ம என்ன சொல்லணும், ஆல்ரெடி சளியை தான் நம்ம என்ன சொல்லுவோம்னா ஸ்பூட்டம். நம்ம இரும பேர் சளியை துப்புறோம்ல, அந்த சளியை தான் நம்ம என்ன சொல்றோம், ஸ்பூட்டம் அப்படின்னு சொல்றோம். ஓகே. we get during the running nose. Running nose ல வந்து அதான் கன்சிஸ்டன்சி இட் வேரியஸ் ஆக்சுவலி. ஒரு மாதிரி எல்லோ விஷயம் ஒயிட் விஷயம் வந்து நம்ம மூக்குல இருந்தும் வரலாம், இல்ல வாயில இருந்தும் வரலாம். பட் அப்படி எக்ஸாஜரேட் பண்ணி வெளியில வர இரும்புற பின்னாடி வெளியில வர்றதுக்கு பேர் தட் இஸ் ஆக்சுவலி. அது ஆக்சுவலி லிக்விடாவும் இருக்கும், அது இன்னும் கொஞ்சம் இதுவாகும் போது கன்சிஸ்டன்சி மாறும் போது கொஞ்சம் சாலிடாவும் இருக்கும். Okay, but both are sputum, but both consist of mucus. Don't confuse sputum mucus. Sputum la irukra oru chinna substance peru da mucus. Okay, right. So we differentiated the locomotion and movement. First we'll start about the types of movement. So when we talk about the movement is of actually, like, we have like two types of movement actually. Non-muscular movement. Non-muscular movement. Other one is muscular movement. So because I already given a break with 5-10 minutes, I'm a break poitanala, so we don't expect break again, because maybe or after just 30 minutes catch a 5 minutes break I give. Okay, so don't expect break, because we'll begin with the next chapter. Okay, so non-muscular movement and muscular movement. As far as non-muscular movement enna type, it can be classified into amoeboid movement, or we have a ciliary movement, and one more we have flagellar movement. These are three types, all the three which comes under non-muscular movement. So amoeboid movement, ciliary movement, flagellar movement. So amoeboid movement, for example, you take like most of the macrophages, WBC, macrophages, the many blood cells if you would have noticed, like today we have class till at 4:45 till 4:45. Hopefully we have a class. Okay, don't worry, I'll be giving another 5 or 10 minutes break for you. Okay, fine. Amoeboid movement, WBC, macrophages. These macrophages are WBC, which shows like amoeboid movement. Fine. So this will take up the cell. You would have studied about like the phagocytosis mechanisms of the WBC, which engulfs the foreign substances by means of the lysozyme, and it forms lysosome. You would have studied in the phagocytosis, right? So this is the mechanism where like phagocytosis mechanism takes place. This is a classical example for amoeboid movement, but this is non-muscular movement. There you could not see any type of, it's a movement, but there you could not see any sort of muscles. So amoeboid movement examples, if you take, you get WBC and macrophages. Then ciliary movement. In case like ciliary movement, for example, ciliary movement where you could see the ciliary movement, our body length ciliary movement in the tracheal way, in the like pharynx, that you could see like you could see the ciliary movement. And also you could see the female reproductive tract, you could see the fallopian tube, right? If you would have studied in the fallopian tube how the fertilization takes place, like how the zygote, 2-cell, 4-cell stage, all these things. This is a short form of macrophage. WBC, white blood cell, and MCM, the very what a phage in macrophage. So for your understanding, I'll write it is a macrophage. The symbol is the phage in. Okay. So ciliary movement you take like fallopian tube. Humans, because we're talking about human physiology, right? So fallopian tube, even in the respiratory tract, in the like pharynx, that you could see the ciliary movement, like this is how the ciliary movement takes place. For organisms, you take ciliates, like paramecium, you take balantidium. These are some of the ciliates even that can also show the ciliary movement, but there are no specialized muscles which playing a important role there. Fine. Flagellar movement examples you take in case of most of the flagellates you studied in the like classifications, like biological classifications. Like flagellar movement la enna neenga padichirukeenga, trypanosoma, trypanosomes, euglena, all the flagellates which comes under the flagella. So And also humans length of flagellar movement, for example, you take a sperm exactly. Like you take an example like sperm, which shows the motility in the female reproductive tract and also in the water, where the sperms, the human sperms can locomote, I mean move faster. This is because of the flagellate structure which is present. So amoeboid movement, ciliary movement, sometimes questions which among the following is a ciliary movement, flagellary movement or amoeboid movement. With all these three which comes under a non-muscular movement. Coming to the muscular movement. So coming to the muscular movement, we have the muscular movement in the lower organisms. Before we going to the human beings, அதுக்கு முன்னாடி muscular movementல வந்து there are other few organisms also which shows a muscular movement. For example, you take hydra பொறுத்த வரைக்கும், if you take like what type of so dinoflagellates கேட்டிருக்கீங்க. What are dinoflagellates? Dinoflagellates are usually algae. It's a powerful mycotoxin it produces. That may not, there are certain very few type of dinoflagellates have, it's actually flagella, but that is also flagellate. That's an algal group. Okay, but dinoflagellates நம்ம சொல்லும்போது நம்ம இதுக்கு மைண்ட் ஃபர்ஸ்ட் வரதே வந்து டாக்ஸின், algal toxin that is produced actually dinoflagellates. Okay, muscular movement of hand, leg, jaw, tongue, இதெல்லாம் வந்து இதுல வரும், muscular movementல வரும். Muscular, now so far we discussed is all about the non-muscular movement. Coming to the muscular movement, before going to the human beings, we discussed about hydra பொறுத்த வரைக்கும் இதுதான் specific type of movements hydra கிடையாது, because hydra shows looping movement, sliding movement, somersault, gliding, loop. There are all different types of floating. There are the different types of depending upon the conditions, depending upon the adaptive situations. Like hydra shows the different types of movements actually. So hydra does not stuck with only one type of movement. Hydra shows the different types of movement. Trypanosoma it's a type of protozoa. Protozoa you would have studied like Trypanosoma brucei, Trypanosoma cruzi, இதெல்லாம் படிச்சு Chagas disease ன்னு படிச்சிருப்பீங்க. Chagas disease அதெல்லாம் human health and disease வரும்போது we'll discuss in detail. So Trypanosoma is actually a flagellate, it's a protozoa. Okay, Euglena உங்களுக்கு தெரியும் actually. Already you studied about the Euglena. It's also like protozoa. Coming to the hydra shows looping, floating, somersault, different types of movement it shows. When coming to the earthworms, can someone tell me like how earthworm moves? Earthworm what type of special structures like which helps the earthworms to move? Can someone tell me like earthworm? You would have எல்லாருமே பார்த்திருப்பீங்க earthworm மூவ் பண்றது இல்லையா? மழை காலத்துல பாத்தீங்கன்னா earthworms சகஜமா வெளியில வரும். So how do like what helps the earthworms to move? Which structure? There is one specific structure which helps the earthworm, because 11th grade you would have studied about the structure of earthworm. Exactly, setae. Setae is actually, it's not a peristaltic movement, it's a setae. Setae is actually the perforation that you could see in the posterior part, like this is the part which helps the earthworms to move from one place to another. Leech, for example, leech எப்படி move ஆகும்? Can someone tell me like leech எப்படி move ஆகும்? Leech. Leech என்ன? Leech தமிழ் என்ன சொல்வீங்க? Leech, how the leeches move. So can someone tell me like how the leech which move from one place to another? Then starfish எப்படி move ஆகும்? How like in the starfish move from one place to another? So longitudinal or circular muscles, exactly. More specifically, when you talk the longitudinal and circular muscles with setae, this earthworm can move. Leech பாத்தீங்கன்னா it's a sticky insects, right? This has got suckers. லீச்சுக்கு அடியில பாத்தீங்கன்னா அட்டைப்பூச்சின்னு சொல்றோம் இல்லையா, இதுக்கு வந்து what you could see is a சக்கர்ஸ் மாதிரி இருக்கும். இந்த சக்கர்ஸ் என்ன பண்ணா இப்ப லூப் மாதிரி இப்படி பெண்ட் ஆகி என்ன பண்ணுன்னு கேட்டீங்கன்னா லூப் மாதிரி ஃபார்ம் ஆகி நீங்க பாத்துருப்பீங்க அட்டைப்பூச்சி பாத்தீங்கன்னா இப்படி போய் இப்படி போகும் இல்லையா. So this is because of the sucker, and also like this makes the looping kind of movements actually. Starfish do have the spines and ambulectal system, that is water canal system, with specific system that you could see in case of starfish, right? The hydraulic suction irukanaala. So ambulacral system, ambulacral or water canal system. This is like water canal system. It is a specific type of system that you could see in case of like starfish, right? The starfish which move from one place to another. These are the different means like இதுக்கெல்லாம் specific movement, but hydra வந்து it doesn't have any specific movement. இது வந்து எப்படி இருக்குன்னா looping இருக்கும். It may be having a floating. It shows even somersault, diving. different movements வந்து hydra வந்து எப்பவுமே இருக்கும். For example, amoeba. Can someone tell me like amoeba என்ன type of movement எதுல வரும்? muscular movement la varuma, non-muscular movement la varuma. Amoeba usually which comes under like with the help of the contractile vacuoles, like this contracts and moves. Actually, this comes under the non-muscular movement actually. Now I'm going to ask you like we're going to discuss about the human beings because our chapter is about the human beings. We'll start with the human. Iniki we're going to discuss about the muscular system. Tomorrow we'll discuss about the skeletal system. Coming to the muscular system, muscles. As we're talking about the muscles, the muscular system, I'm gonna ask you like the few questions like there are a few types of muscles, like how many approximately how many numbers of like number of muscles are there in your body? Can someone tell me what are the number of total approximate number of muscles? Number of muscle in your number of muscles in your body. Actually, the muscles are made up of actually it's a mesodermal in origin. It's a meso because ectodermal, endodermal, mesodermal அந்த மாதிரி த்ரீ டைப் ஆஃப் லைக் ஆரிஜின் இருக்கு. இட்ஸ் டிவிஷன் ஆஃப் லேபர் நீங்க படிச்சிருப்பீங்க. அதுல மசில்ஸ் வந்து எதுல இருந்து வரும்னு கேட்டீங்கன்னா this comes under the mesodermal in origin. The type of cells, like the special type of cells, the muscles which consist of myocytes. The myocytes எப்படி liverல தான் நீங்க hepatocytes அப்படின்னு சொல்றீங்களோ, the same way the muscles which consist of a specialized type of cells called as myocytes. Now, how many number of muscles actually? So when we talk about the number of muscles, we have 639 muscles. Approximately 639 muscles are there in a body. Out of 639 muscles, there are 317 are paired muscles. The 317 has to be multiplied into 2. So 317 into 2, and the remaining like 5 are unpaired muscles. 5 are unpaired muscles. This is approximate number only, 639. 639 is actually the approximate number where in the 639, 317 muscles are paired muscles. The pair muscles. There are 5 muscles which seems to be unpaired muscles. Okay. Now I'm going to ask questions like which is the strongest muscle. The first question is actually which is the largest muscle in our body? Which is the largest muscle? நம்ம உடம்புல இருக்கிறதுலே பெரிய muscle எது? Which is the largest muscle? You can answer me. Which is the largest muscle in your body? And the next one is the longest muscle. The longest heart muscle. Then we go with the longest. We have the soft muscle. This is like soft, and this is the longest hard muscle. Then we have strongest muscle, which is the strongest muscle, and which is the shortest muscle. So maybe like if you know the answer, you can tell me actually. So I need actually exact the name. Suma, you should not tell like tongue, thigh, and all. Yes, someone tell me like gluteus maximus. Exactly, like gluteus maximus is the largest muscle. So the largest muscle in your body is actually the gluteus maximus. It's a hip. You could see in the hip muscle. Hip. The name of the muscle is gluteus. You need not by heart all the muscle. It's not a compulsory because the textbook they are not given all the names of that muscle. Only these names are sufficient actually. The largest muscle is in the hip region of the human body. The name of the muscle, the largest muscle is the gluteus maximus. Coming to the longest soft muscle. When coming to the longest soft smooth muscle is what? What is the longest smooth muscle? Rectus abdominis. Rectus abdominis. Rectus abdominis is actually the soft or smooth. It's instead of soft, I call it as a smooth also. The longest smooth muscle is the rectus abdominis muscle actually, and which is actually like longest hard muscle, which is at the back, and this is called as sartorius, which is actually the back side of your body, back side, which is at the back. The back side you could see, right? So this is actually a sartorius muscle. This is the longest muscle, but this is the hard muscle that you can see, which is actually the strongest in your body. Which is, can you tell me like which is the strongest muscle? Where you could see the strongest muscle? The strongest muscle which present in your jaw. Can you tell me the name of that muscle? What is the name of the muscle which present in your jaw? Yes, I just waiting for that. So what is the name of the muscle which present in your jaw? And shortest muscle. The shortest muscle, the shortest muscle is stapedius muscle. It's a stapedius in your here part, right? You will study like male ears, incus, and states, like the middle ear, the bones, Very good here. Where you could see the shortest muscle, that is a stapedius muscle, and the jaw. You answered, the one of you answered the correct thing. Is it which is the answer for the strongest muscle which present in the jaw part? Is a masseter. Masseter is actually the name of the muscle. The jaw part, we have masseter. So you needn't by heart all the muscles. Maybe like so many muscles are there. You needn't worry about all the muscles. And you just recollect. Often they ask which is actually the largest muscle. The gluteus maximus is the largest muscle. And masseter. Masseter is actually the jaw part. We have the upper jaw, the jaw, right? This area that you could see the masseter muscle. So, and the shorter muscle is the stapedius. We ask like fallopian tube. So, fallopian tube. No, fallopian tube is actually the part of the female reproductive system where you could see the ciliary movements. So that only I mentioned the fallopian tube. Okay. Right. So these are actually the few names of some of the muscles that you should remember. Okay, masseter is actually the jaw muscles. Can I move on to the next slide? I hope you made note of this. So largest muscle, longest soft muscle, I mean smooth muscle. The longest hard muscle is sartorius muscle, and it's one R here. Sartorius muscle. The strongest muscle is in the jaw, that is the masseter we call it as. And we have the shortest muscle, and that we call it the stapedius. And where you could see the stapedius muscle, we could see the ear ossicles, like what are three bones, the three pairs of bones you could see in your ear, that's malleus, incus, and stapes. In that part, you could see this type of muscle. And this is just for your reference only, so don't worry about the other types of muscles, right? So maybe the question, if at all they ask any questions from the name of the muscles, usually they, okay, can you move to the next slide? Fine. So we move to the next part. The next part is actually the types of the muscles actually. So the types of muscles. When we talk about the types of muscle, we have three types of muscles. You know already, right? What are three types of muscles? We have the three types of muscle. Is skeletal muscle. Skeletal muscle. We have the visceral muscle. Visceral muscle. We have then the cardiac muscle. So we have three types of muscle. But in the textbook, they elaborately, they discuss only about the skeletal muscles because we're going to discuss only about the skeletal muscles today. But you need to understand what are the other two types of muscles. So other type are the visceral muscle and the cardiac muscle. So what makes the difference between these three types of muscle? These the skeletal muscles are actually striated. You could see some lines on its surface. So we call it as a striated. And visceral muscle. Visceral means what all the things that inside our organs, like inside our body, right? All the internal organs made up of certain muscles, right? And these muscles are called as organs like liver, kidney, like all other like internal organs, intestine, all these are called this type of muscles. That is visceral, not visceral, it's a visceral inside, which is present inside. These type of muscles are unstriated muscle, cardiac muscle. Cardiac, especially for the heart, it is a striated muscle. So as far as visceral means organs, internal organ. So if I mention about the internal organ, I use the word called as visceral muscle. So how will I differentiate? So skeletal muscle is striated. Striated means I could see the line. For example, if I talk like this is a striated muscle, I could see this type of kind of the lines that roll the muscle. I could see this type of the lines of striations I could see on a surface, but that's why I call it striated. You could see the visceral muscles. Visceral muscles usually you'll be having this type of arrangements. Here you could see the central cell, but the tapering ends. You could see the tapering ends in case of the visceral muscle, but here you could not see any sort of striations. So I mentioned this as an unstriated muscle. Okay, but in case of like cardiac muscle, here you could see like cardiac muscle. Here you could see like branch like structures that you could see in case of the cardiac muscle. Here also you could see striations. Here also you could see the striations. Like striations I could see both in a cardiac muscle as well as in the skeletal muscles. Both the cases I could see the striations. And as far as like an action is concerned, skeletal muscles are voluntary in action. It is voluntary in action. Visceral muscle. Can you tell me like visceral muscles are under your control? Can you control your kidney? Can you control your like internal organs? No, definitely. Because the reason is these muscles, the visceral muscles, are involuntary in action because you cannot control these muscles. So these muscles are involuntary in action. Coming to the cardiac muscle, again, can you control the heartbeat? If the God has given you privilege to control, I mean, muscles under your control means for time being you control your heartbeat, then you may not survive. You may forget to, like, make the heart work again. So it's again cardiac muscles again involuntary in action. This is not under your control. So you can control only the skeletal muscles. You can increase your, make a biceps, you make a six pack, you can do whatever things that you want. That all things that you do because it is under your control. That is your skeletal muscles, right? But your visceral muscles are involuntary in action, and cardiac, the heart muscles, right, that is also like involuntary in action because both these muscles are not under your control, so it is involuntary in action. So this is for your thing. And coming to this, like, here it is, could see the uninucleated. Here it is like, uninucleated. Here it is like multinucleated, you could see. Then here it is uninucleated structures. So this is how actually you can make out the difference of three types of muscles. This is now, but this is unbranched. Skeletal muscles are not like branch, right? I just gave an example like six packs. So if you go for like gym and all, like, guys, you craze about, like, making like six packs and all. Why are we getting six packs? Because six packs, that is because you're working out, right? You're going to gym and working out. That is the reason, because it is voluntary in action. Since you're working on it, you're controlling it, you're making that to happen. So that is the reason I told you like six packs. So don't write out six packs and all this. I just gave an example. Okay. Okay. So how they are formed, what happens actually, and how they are formed. So you asked this good question, but we're going to discuss how this conditioning, how this, like, the muscles are getting, because the main function of the muscles like elasticity, excitability, then contraction and relaxation. These are the main four functions of the muscles. Because what are the four main functions of the muscles? Contraction, relaxation, excitability, right? So these are the main functions of the muscles actually. Elasticity. These are the main four functions. If you say about the six packs or what you're making a body fit, means it means you're working out. The reason, one thing that you should understand scientifically is skeletal muscles are voluntary muscles, but other two types of muscles like visceral muscles and cardiac muscles are involuntary in action. Okay, now, and this is like unbranched. One more thing you can make here is like, it is a unbranched, and here it is branched. Actually, cardiac muscles are branched in structure, right? So here you can make out the difference. Now we discuss in detail about the skeletal muscles. If we're talking about the skeletal muscles, for example, if you draw the structure called as, like, this is actually the bone, and here I'm talking about the muscle. This is actually the muscle I'm talking about. So the skeletal muscle. I'm talking about the skeletal muscle. So what is actually the connective structure? This is a connective structure that you could see. What connects your bone and your muscle? Can someone tell me, like, what is the connective structure we call it as? This is actually your bone, and this is your skeletal muscle. This is a skeletal muscle. Okay, there is something, because your muscle don't have a privilege of having a direct connection with your bone. This bone has got something in between, the bridge kind of stuff which is happening in between. So what is actually, it's a ligament. There is some other name, tendon, exactly. So answer, tendon. So this structure is called as tendon. So the bone is getting connected with the skeletal muscle by means of tendon. Now I just going to tell you, like, how this is the structures exactly look like. So maybe like the image representation which tells you. I hope this is visible for you. So if you take the structure, this image will tell you, like, the structure of so if you make the structure, this is how actually the muscle, if you, this is the hand, the part that you're talking about, right? So how the muscles from the tendon, this is the tendon part I'm talking about. From the tendon, we have the bundle. We have the bundle kind of structure which emerges out of the, this is actually the bone. This is actually the bone part that you could see. This bone is actually is getting connected with, this is, I mean, tendon I'm talking about. And this is where actually the muscle comes. Okay. Okay. Expand. Which part you're asking for? Expand. Expand IT. So this is actually the muscle, the fiber. See, you see the section of the skeletal muscle. Here the skeletal muscles consist of, see, here you could see the numerous bundle-like structure you could see, right? This bundle which emerges from fascia, this bundle is called as a complete bundle. With that we call it as, I'm talking about the muscle. We started with the muscle now. Okay, skeletal muscle. Don't think about a cardiac muscle and visceral muscle because that is not given, I mean, explained in our textbook. We're talking about only the skeletal muscle. Fine. As far as skeletal muscle is concerned, because what is the feature of skeletal muscle? It's a involuntary. It is like voluntary in action, and also it is having a these muscles are having a striations. Now you could see the fascium. Okay, this fascium is a structure. Inside you could see like numerous bundles actually. So this bundle will be having like structures like this inside. Like you could see the structures like this, which will be coming out the fascium. Like this, you have the bundles which will be coming. So this is actually we call it as a muscle fiber. This is called as muscle fiber. Yeah, this is a muscle fiber. In the muscle fiber, so you have something. This is a muscle fiber. In the muscle fiber, there is actually like the outermost structure, like this is a sheath kind of arrangement. Inside, you'll be having something called again. This will be going to minute fibrils, like which will be coming from inside. So minute structures which will be coming from inside. This structure is actually we call it as a myofibril. This is called as myofibril. Okay. So from the myofibril, so again from the myofibril, even you can go deep inside. It's a bundle like you could see in this part again. It's going inside. Like you could see like again the bundle of muscle cells. You could fascicles, and in the muscle cell consists of many myo. This is the part I'm talking about here. This is actually the myofibrils and talking this myofibrils here. You see the myofibrils here, right? Now you take even deeper, go deeper inside the myofibril. If you take the myofibril, the outermost structure is actually we call it as sarcolemma. This is called as sarcolemma. The sarcolemma, which consists of the inside the fluid, is called as sarcoplasm. Inside the fluid which is present inside is called as sarcoplasm. The sarcoplasm consists of sarcoplasmic reticulum. We call it as inside the fluid. We have something called as SR. SR stands for sarcoplasmic reticulum. Okay. Now inside the sarcoplasmic reticulum, we have the other like this individual unit which is coming out is actually we call it as a this unit is called as sarcomere. This is called as sarcomere. So what is the structural and functional unit of the muscle? Is the contractile unit of the muscle is a sarcomere. Okay. So the question comes, what is actually the contractile unit? So you can contraction, relaxation, both are playing important role, but sarcomere are playing very important role in the contraction of the muscles actually. Okay. So the unit, which is actually the functional and structural unit of the muscles, is actually the sarcomere. We need to zoom in and just go deeper inside. This diagram, this image, like explained clearly. From the muscle, you could see the fasciculus. The fasciculus, which again rooted to the bundle. In the bundle, which consists of the again the myofibril. The myofibrils, which consist of the sarcomere. So in deeper, if you go inside, like you have a contractile unit. This contractile unit is called as sarcomere actually. So if you go to the structure, this is what actually talking about the sarcomere. So if you take the sarcomere, the section of the longitudinal structure of the sarcomere or under the electron microscope. If you see under the electron microscope, this is how the structure of the sarcomere look like. Now I'm going to tell you like what are the different bands or different regions that you could see in the sarcomere. Like this, you'll be having so many sarcomeres. If you take the one muscles, like you'll be having repeated sarcomeres. You'll be having a repeated sarcomeres. For your understanding, for explanation purpose or teaching purpose, I just took just a single sarcomere. I'm going to tell you like what a single sarcomere consists of, what exactly is happening inside this sarcomere, what makes the contraction and relaxation. Once we're done with this part, then you can understand the remaining things like the proteins like actin, myosin, troponin, tropomyosin, all these things that you can discuss. Now we move on to the structure of the sarcomere. When you go to the structure of the sarcomere, when you go to the structure of the sarcomere, so sarcomere structure. You take the sarcomere structure. As far as the sarcomere structure is concerned, so before going to sarcomere structures, we have the muscle fibers. I told you like muscle fibers. There are two types of muscle fiber. Can you tell me like what are the two types of muscle fiber? There are two types of muscle fiber. So the muscle fibers are of two types. What are the two types of muscle fiber? One is red muscle fiber, red muscle fiber. Other one is white muscle fiber. So there are two types of fiber because human body we consist of both the types of muscle fiber. RMF means red muscle fiber, and WMF stands for white muscle fiber. There are few differences that I can enlist between RMF and WMF. RMF is thin. White muscle fibers are thick in nature. The next difference that you could see is actually there are more number of myocytes here, more number of myocytes. Here it may be absent. The myocytes are absent, or it may be very less. Okay, a very negligible amount, very less, or sometimes it may not have this type of myocytes. That way also you can differentiate like right and white muscle fiber. The other thing is contraction. Here in red muscle fiber, the contraction which takes place very slower. Here, very faster rate. The faster rate you could see like white muscle fiber contraction takes place. Then, example, if you take the red muscle fiber, here you could see backside. Your backside muscles, backside backside muscle. At the back, you could see the backside muscles is example for the red muscle fiber. Can you tell me where exactly, any one place, can you see, tell me where the white muscle fibers are present? I told you like, the things like red muscle fiber, that is like backside, the backside of your muscle. Can you tell me one example? Because I told you already, like, here the contraction takes place much faster. Can you tell me one particular place in our body? Because you cannot strain that particular muscles for a long span of time, because within the short span of time, like the muscles which go for a contraction, and you feel like irritation and you feel like a sense of discomfort. Can you tell me which part of the body, especially like which organ, has got like this type of white muscle fiber? So you can just find out and tell me before we close the session. So before going to the sarcomere structure, you need to understand the difference between red muscle fiber and the white muscle fiber. Okay, so this is okay. Our RMF is red muscle fiber and WMF is a white muscle fiber, because it's not like textbook, like they're not clearly described or like differentiated between RMF and WMF, but you need to know what is the difference between the muscle fibers. Now we move on to the sarcomere structure. Coming to the sarcomere structures, we have like two type of bands actually. One is A band, the other one is I band. So we have two type of band, A band and I band. A band stands for anisotropic band. So you answer high, exactly like your eyeballs, right? Example, you answered me correctly, like eyeballs has got white muscle fiber. That is the reason, like why if you sit in the front of a computer, you operate the phone and looking at the phone for a long span of time, like immediately you got reddening of eyes, it started irritating, right? This is because of this white muscle fiber, because the contraction takes place much faster. Eyeballs, like white muscles that you could see in the eyeballs is the correct example for the white muscle fiber. So A stands for anisotropic band and I stands for isotropic band. Okay. So A and I. So what A band, which consists of A band, which is actually otherwise called as, it's a dark band actually. It's a dark, and I is actually otherwise called as a light band. A stands for anisotropic band. Anisotropic band. I stands for isotropic band. I stands for isotropic band. It's a light band and a dark band. So what the A band consists of? A band consists of a protein called as myosin. I band, which consists of protein called as actin. Okay, which consists of an actin. Okay, isotropic band. So here this consists of I bands, and why it's light because of the actin protein. Here is a dark, it is because of the myosin protein. If I make the diagrammatic representation of the previous slide, I just shown on slide, right? So for your better understanding, I just draw and explain like how these bands, I mean the sarcomeres look like. For example, this is actually, I take it as a band. This is a A band. In between, you could see something called as high band. It seems to be overlapping. It seems to be overlapping. like this. You could see A band and you could see the I band. So this part, the shaded part, right? This shaded part is actually, let me use like other color for your reference. So you could see this red region, right? This red region is actually for all these are actin. These are all actin, and this is what actually call it as like eye region. And this one like central part is actually we call it as a band. So now if you see, this is actually this complete part. This part is actually the complete part. Central part is actually the full part is actually call it as a band. This is actually call it as A. And this region, right, the red region, the red shaded region, and this red shaded regions actually we call it as I band. So this red region here from here to here, this is actually we call it as I. This complete part, the middle part is A, and this overlapping the red shaded region that we call it as I. Understand, like this consists of the central part, which A, it consists of myosin, and the red region, this overlapping, right, this consists of the actin. This I band, A band, this present in the sarcomere. Where the sarcomere is present? Myofibril. Where the myofibril is present, it is in sarcolemma. Where the sarcolemma is present, it is present in the muscle fiber. Where the muscle fiber is present, it's present in the fascium. Or where the fascium is present, it's a skeletal muscle. Getting what I mean? We are talking about a small unit, the structural and functional unit is actually sarcomere. In deep inside, when you just go zoom in and deep inside, you could see something called as a functional unit sarcomere. It's just talking about just a single unit in the muscle, that is in the myofibril. That is actually, these are all present in the, where it is present, it is present in the muscle, actually in the sarcomere. You could see this bands actually. Next, this we have like an A. This is actually A, and we have something called as in the A region we have something called as like this is, I told you, the dark region, right? In the dark region we have something called as this, something called as like the line that you could say, make it as a line. This is called as H line. This green color is actually called it as a H line. This is called as H line. Where the H line is present or H zone is present, H zone is present in the A band. What A stands for? A is a dark band. In the dark band, there are some light bands are also present. Some in between, like you could see some light regions are there, right? Light band is present. This light region is actually, we call it as a H zone. Okay. Now, H zone again, like in between, it consists of like the other part, that is, there's a line. This is actually the central part. We call it as a M line. In between this H line, we have a M line. M line. So maybe a little bit it is like confusing part. I'll just explain it. See here, we have something called as like this is central part is a A band, overlapping part is actually I band. A is for the dark, I is for the light. Okay, central part is a dark and overlapping part is a light. In the dark part we have some light region as well. That light region is otherwise called as H line. Okay, that is called as H line. In the area மட்டும் கொஞ்சம் லைட்டா இருக்கும். அதாவது டார்க் பேண்ட்ல சில ஏரியா வந்து லைட்டா இருக்கும். அந்த லைட்டான ரீஜன்ஸ் நம்ம என்ன சொல்றோம்னா H ன்னு சொல்லுவோம். அந்த ரெண்டு H லைன்ஸ்க்கு நடுவுல இன்னொரு சின்ன ஒரு லைன் போகும். That is actually we call it as a median line, or we otherwise called as M line. Getting? So far we discussed only about like A band, I band. Then we discuss in the A band we have the H line and we have a M line. Okay. So now we talk about, there is one more line that is present. Actually, we call it as a Z line. We call that line as a Z line. So here you could see the Z line. This place you could see the Z line. The where the Z line is present, the Z line or Z line is present in the this region, I band. So this is actually the part we could see the Z line. Apo, what is actually the sarcomere in the Z line? This is actually Z line. This is actually Z line. This from year to year, from in the Z line learn in the Z line learn, we call it as a sarcomere. So what is a sarcomere? Sarcomere is a unit which present between two successive Z line. இந்த Z line को இந்த Z lines को நடுவுல இருக்கிற அத்தனை ரீஜனுமே நம்ம என்ன சொல்றோம்னா we call it as a sarcomere. இதே மாதிரி அடுத்து we could see like repeated sarcomeres are present throughout the மயோஃபைப்ரல்ஸ். இதெல்லாம் எதுல இருக்கு? மயோஃபைப்ரல்ஸ்ல இருக்கும். நான் வந்து உங்களுக்கு ஒரு புரியறதுக்காக ஜஸ்ட் ஒரே சின்ன செக்மெண்ட் மட்டும் சொல்லியிருக்கேன். சோ உங்க மசில்ஸ் வந்து கான்ட்ராக்ட் ஆகுது, நீங்க ஜிம்முக்கு போறீங்க, யூ கான்ட்ராக்டிங், ரிலாக்ஸிங், யூ டூயிங் எக்ஸசைசஸ், யூ டூயிங் டேக்கிங் புஷ்அப்ஸ் அண்ட் எவ்ரிதிங், யூ வார்ம் அப் யுவர் பாடி. இதெல்லாம் எப்படி அப்படின்னு கேட்டீங்கன்னா, திஸ் இஸ் பிகாஸ் ஆஃப் ஒரு யூனிட், லைக் ஒரு ஜஸ்ட் ஒரு மசில் ஃபைபர்ல இருக்கிற ஒரு சார்கோலிமால இருக்கிற ஒரு மயோஃபைப்ரல்ல இருக்கிற மயோஃபைப்ரலுக்குள்ள இருக்கிற ஒரு ஜஸ்ட் ஒரு சின்ன பார்ட், சார்கோமியர் என்ற பார்ட். இந்த சார்கோமியர்ல இந்த மாதிரி ஒரு ஸ்ட்ரக்சர் இருக்கு. திஸ் கோயிங் டு பிளே வெரி இம்பார்ட்டன்ட் ரோல் இன் தி கான்ட்ராக்ஷன் ரிலாக்ஸேஷன். தட் ஆல்சோ சார்கோமியர் வந்து கான்ட்ராக்ஷன் ரிலாக்ஸேஷன் ரெண்டுமே பண்ணுமா? இல்ல. பிகாஸ் சார்கோமியர் இஸ் கால்ட் அஸ் contractile unit. Question வந்து sarcomere is a structural functional unit of the muscle, correct? But sarcomere is actually playing very important role in the contraction. So that's the reason is அப்ப relaxation யார் பண்ணுவா? Relaxation is performed by, actually there's something called as T tubules னு ஒரு tubules இருக்கும். So where is the T tubules are present? The T tubules are present here in this case. Z line is சுத்தி, for example, the Z lines are surrounded by the tubule like structure. இந்த Z line ஐயும் இங்க இருக்க Z line அப்படின்னா full sarcomere. நான் ஆல்ரெடி சொல்லியிருக்கேன், what is the sarcomere? இந்த Z lineல இருந்து இந்த Z lineல இருந்து full region நம்ம sarcomere சொல்றோம். அப்போ this Z line is also surrounded by T tubules. This white tubules is called as T tubules. T னா transverse tubules னு சொல்லுவோம். இது இங்க T tubules இருக்கும், இங்க என்ன இருக்கு? Transverse tubules இருக்கும். So this T tubules along with the sarcoplasmic reticulum. So T tubule plus sarcoplasmic reticulum plus calcium ions. இது மூணுமே சேர்ந்து தான் உங்களுக்கு என்ன பண்ண போகுதுன்னு கேட்டீங்கன்னா ரிலாக்ஸேஷனும் கான்ட்ராக்ஷனும் பண்ண போகுது. So contraction, relaxation both are going to take place with the help of the T tubules. So where the T tubules beginning and end. The full sarcomere beginningல இருக்கும், endல இருக்கும். ரெண்டு இடத்துலயுமே வந்து the Z line is entirely covered by the tubule-like structure called as transverse tubules or the T tubules. ஏன் இது வந்து ரிலாக்ஸேஷனும் கான்ட்ராக்ஷன் பண்ணுதுன்னு கேட்டீங்கன்னா, நர்வ்ஸ்ல இருந்து பிரெயின்ல இருந்து வர இம்பல்சஸ் இருக்கு பாருங்க, சென்ட்ரல் நர்வஸ் சிஸ்டம்ல இருந்து வர இம்பல்சஸ். சென்ட்ரல் நர்வஸ் சிஸ்டம்ஸ் என்ன ப்ரொடியூஸ் பண்ணும்? இம்பல்ஸ் ப்ரொடியூஸ் பண்ணும். இம்பல்ஸ் நேரம் எங்க வரும்? நியூரோ மஸ்குலர் ஜங்ஷனுக்கு வரும். நியூரோ மஸ்குலர் ஜங்ஷன் வந்து அங்க இருந்து this will be coming to your neuron. Neuron எந்த ரீஜனுக்கு வரும்? ஆக்சான் வரும். ஆக்சான்ல எந்த ரீஜன்? சினாப்டிக் கிளெஃப்ட்ல வரும். சினாப்டிக் கிளெஃப்ட்ல என்ன ப்ரொடியூஸ் ஆகும்? அசிட்டில்கோலின் ப்ரொடியூஸ் ஆகும். This acetylcholine which will come and reach to this region, that is actually like to the muscle, a small unit of the muscle க்கு வந்து சேரும். எங்க? the T tubulesல வந்து சேரும். So the signals are getting passed from the central nervous system. It's keep on passing to your T tubules. Now the T tubules are responsible for the contraction and relaxation. Already I told you sarcomere is the contractile unit. Relaxation சேர்த்து பண்றது யாருன்னு கேட்டீங்கன்னா only this T tubule. So இந்த மாதிரி just ஒரு யூனிட் வந்து இந்த மாதிரி நடக்கும். அதே மாதிரி அடுத்து லைன் என்ன இருக்கும்னு கேட்டீங்கன்னா உங்களோட மயோஃபைபிள்ல இந்த மாதிரி sarcomeres தான் இருக்கும். அப்போ ஒவ்வொரு யூனிட்டும் வந்து, for example, you're contracting your muscle like this. Then what is happening, like this T tubules உங்க central nervous systemல இருந்து உங்களுக்கு signals pass ஆகுது. How? Neurotransmitters like acetylcholine மூலியமா என்ன இருக்குன்னா messages pass ஆயிட்டு இருக்கனால this is going to this particular region and automatically like T tubules is going for relaxation and contraction. So this is how actually your sarcomere is function. உங்க ஒரு மசில் வந்து கான்ட்ராக்ட் ஆகுது, ஒரு மசில் வந்து ரிலாக்ஸ் ஆகுது, நீங்க உட்கார்ந்து எழுந்து whatever the job that you're doing, even getting up from the bed and just sitting and watching this video, even for this, right, you're blinking your eyes, but that is also like some muscles supposed to go for contraction relaxation. Don't think like vigorous exercise பண்ணாதான் contraction கிடையாது. You just get up from the bed, you blink your eyes, you smile, you frown your eyes, நீ என்ன வேலை பண்ணாலும் you're doing something, right? The movement is happening. This is happening to the skeletal muscles because of this sarcomere unit. This sarcomere unit, especially the T tubules, don't come to a conclusion sarcomere is playing the relaxation. Sarcomere is only the contractive unit, whereas the T tubules, which is like present on both the sides of the sarcomeres covering the Z line, this receive the impulses, receive the signals like acetylcholine, and this makes some particular unit or the muscles to go for contraction and relaxation. At contraction, whether the width of A band changes, we're going to explain. Next one, we're going to discuss about the mechanism. எதை வச்சு நம்ம muscle contract ஆகுதா ரிலாக்ஸ் ஆகுதான்னு கண்டுபிடிப்போம்னு கேட்டீங்கன்னா, இந்த zone இருக்கு பாத்தீங்களா, this is called as H line சொல்றோம்ல, இந்த H line தான் என்ன சொல்றது, I call it as a H zone. இப்போ இந்த கேப் இருக்கு பாருங்க, you could see in the green line, this green line in between space நிறைய இருந்துச்சு அப்படின்னு கேட்டீங்கன்னா the muscle is in relaxed condition. அதே வந்து ஃபார் எக்ஸாம்பிள் ரெண்டு எட் ஜோன் வந்து ரொம்ப கிட்ட இருக்கு, ரொம்ப ஸ்பேஸ் கம்மியா இருக்குன்னா the muscle is in relaxed condition, I mean like contractive condition. So based on the gap of the egg zone இருக்கிற gap பொறுத்து தான் you can come to conclusion whether the muscle is in contraction or the muscle is in relaxed. இப்போ z line ஒரு z line இங்க இருக்கு, இங்க கேப் பெருசா இருக்குன்னா அப்படின்னா மசில் வந்து ரிலாக்ஸ் ஸ்பேஸ்ல இருக்குன்னு அர்த்தம். இப்போ if you doing exercise அப்படின்னா கண்டிப்பா நம்ம மசில்ஸ் என்ன ஆயிருக்கும், எட் ஜோன் வந்து கிட்ட வந்திருக்கும், இந்த ரெண்டு லைனும் கிட்ட வரும்போது the muscle is in contractile condition. So based on the looking at the gap of the edge zone, we conclude like whether the muscle is in contraction or is in a relaxation. So in this screen shows like the structure of sarcomere. Coming to the sarcomere, I just give like quick things. What are things that you should remember, because it's like clumsy, the board seems to be clumsy. I tell you like sarcomere structure, the dark band and the light band. A வந்து dark, I வந்து light. Central part is actually we call it as A band. Overlapping red color regions that we call it as a I band, right? In the A band, you could see some light region as well. That light region is called as H line, okay, or H zone. In between the H zone, we have some median line. This median line, the pink color is called as M line, okay. And on the actin, this part, right, the light band, you could see like the orange color, like the line that I drew. This is actually we call it as a Z line. So the difference between the gap which takes between two Z line is called as sarcomere. Now the Z line which covers by the tubules, like transverse tubules, and this create like this receives the signals to acetylcholine, and this is responsible for the contraction and relaxation of the muscles actually. And I give you like the other name, the Z and then keeping it Z. So Z க்கு so Z line. So what is actually the Z line? Z line is called as Zwischen sheep. இது கொஞ்சம் டிஃபரெண்டா இருக்கும் pronounce பண்றதுக்கு. Z line is called as Zwischen sheep. This was actually found by this particular person, the scientist name is Zwischen sheep. The Zwischen sheep is actually like meant for a Z. The Z into the Zwischen sheep. What is the other name for the Z line? The Z line can be otherwise called as Dobie line. Dobie நானும் Z line தான் அர்த்தம். Cross line, Krause, cross or Dobie or Zwischen. So all these three different names that we give for the Z line. Sometimes Zwischen கொடுக்கல, Dobie or cross line அப்படின்னு கொடுத்தாங்கன்னா it means like they're talking about only the Z line only. So why is that? What's the significance of line? Z line is nothing but the difference actually. இங்க இருக்க Z line க்கு இங்க இருக்க Z line க்கு நடுவுல இருக்கிறது தான் we call it as a gap, that is we call it as a sarcomere actually. So this shows the structure of the sarcomere. Now we move on to the, you can ask me like what is actually the size of the sarcomere? ஒரு sarcomere ஓட size எவ்வளவு சார் இருக்கும்? So the size of the sarcomere is actually 2 to 3 microns. 2 to 3 microns. Often they ask this question. The size of the ஒரு sarcomere ஓட size, ஒரு sarcomere ஓட size வந்து என்னன்னு கேட்டீங்கன்னா is actually 2 to 3. Sarcoplasmic reticulum and sarcolemma. Sarcolemma is actually the layer, is a layer, and the layerல இருக்கிற actually the perforations are called as sarcoplasmic reticulum. Both are not the same. So isotropic into the getting iso means it's actually the light, same sort of like proteins that you could see is in this case like isotropic, it's actually light, actually it's a light. When you stain it, this gives actually light color, lighter. So we call it as a, it takes up light color. அதே வந்து நீங்க anisotropic அப்படி பாத்தீங்கன்னா which takes up the stain and looks in a darkened color. So this is isotropic and this one is anisotropic. Isotropic இருக்கிற protein பேர் தான் we call it as an actin, that means light. Anisotropic வந்து A band, it means the name of the protein which is present in the A band is actually myosin. Okay, so surrounds the Z line. The thing which surrounds the Z line is the T tubules. T tubules அப்படின்னா it's not just a Z line, full sarcomere. இந்த full sarcomere வந்து இந்த cross ஆன வரைஞ்சிருக்கனால Z line சுத்தி போட்டுருக்கேன். It means இந்த Z line க்கு Z line க்கு நடுவுல இருக்கிறது தான் sarcomere னு சொல்றோம். அப்படின்னா this covers the entire sarcomere. Acetylcholine signals வரும்போது it should pass to the entire sarcomere. So it covers the entire sarcomere. It means Z line. Z line which is actually the beginning and the end of the sarcomere. அதை represent பண்றதுக்கு தான் உங்களுக்கு Z line சுத்தி இருக்கிற மாதிரி போட்டுருக்கேன். So this covers entirely your Z line. So this is about the structure of sarcomere. So time is like 4:30. Even if I give a break also because I got off completing by 4:45. So kindly bear with me for another 15 minutes. I try to complete like another just one subtopic. If I give a break again, then again like it will be extended till 5 o'clock. So I hope you guys can retain another for another 15 minutes. Okay. We go to the next part. Sarcomere structure we discussed. Next we discussed about the proteins actually. What are the proteins? The molecular structure. Okay, we discussed about the sarcomere part. Sarcomerல ஆக்டின் மயோசின் அப்படின்றத பார்த்தோம். ஆக்டின் மயோசின்றது நம்ம சொல்ற பேர், பட் அதுக்குள்ள என்ன இருக்கும்? இட் கன்சிஸ்ட் ஆஃப் சம் புரோட்டீன் இல்லையா? ஸோ அந்த புரோட்டீனை பத்தி we going to discuss. We discussed about the proteins which present in what? Present in the this particular sarcomere. So molecular structure. So we did நம்ம ஃபர்ஸ்ட் பார்த்தது வந்து general structure. Now we going to discuss about the molecular structure of the sarcomere. Come to the molecular structure, we have four types of protein. In the four types of protein, எப்படி பிரிக்கலாம் அப்படின்னு கேட்டீங்கன்னா, we differentiate into two types actually. Contractile protein, regulatory protein. Contractile protein, regulatory protein, and there's something called as key protein கூட சொல்லலாம். இந்த மாதிரி மூணு type of proteins இருக்கு actually. ரைட்டா? Contractile protein. Contractile protein which consists of actin. இன்னொரு protein யாரா சொல்லுங்க பாப்போம். What is other protein? I make it as a blank. You tell me what is the other protein. You listen to this class. Even the last இதெல்லாம் clippingல சொன்ன உங்களுக்கு இன்னொரு protein என்னன்னு சொல்லிட்டு. Okay. Regulatory protein. So regulatory protein அப்படின்றதுனா here in this case regulatory protein we have troponin. Another protein enna that you can tell me what is the other protein you could see along with the troponin? Yes, you are right. Myosin. Myosin is other protein. Troponin or protein. What is the other name? What is the other protein which is present along with the troponin? That you have to just tell me like what is the other protein. Coming to the key protein. Coming to the key protein, we have titin. Then you would have not seen in the books actually. Still other proteins are good involved actually. So the proteins are actually the titin. Then we have myomycin. Myomycin. Then we have dystrophin. Dystrophin. These are all the additional key proteins. Okay. Troponin, tropomyosin. Good. So there are three types of proteins which present in the sarcomere. Contractile protein, regulatory protein, and key protein. Key protein la வந்து titin இருக்கு, myomycin இருக்கும், and we have the dystrophin gene actually. Can you tell me like one important function, ஏதாவது ஒரு molecular aspect dystrophin ஓட main feature, அதாவது you just find out and tell me like what is the main feature based on its molecular weight or something related to the molecular biology, like what is the main feature of this particular protein? It's a protein actually, dystrophin. What is the main feature, the molecular aspect of dystrophin? You just tell me. Okay, so these are the three types of proteins. First we'll discuss about the myosin protein. As far as the myosin protein is concerned, this myosin can be treated with trypsin. Trypsin which you treat பண்ணும்போது, when you treat with the trypsin, this can be divided into the two segments of fragments of period. One வந்து light meromyosin, other one is heavy meromyosin. So these two fragments that you'll get when you treat this particular protein called as myosin with an enzyme like trypsin. Trypsin which treat பண்ணும்போது, you'll get two segments actually. These two segments, light meromyosin and heavy meromyosin. இந்த மாதிரி ரெண்டு விதமான இது கிடைக்கும். When you look at the structure of this myosin, look at the structure of myosin. So you get a structure like this, for example, double structures in the mari, you get the double structure and you get the structure like this. So it's a double structure actually, and this consists of like this is a head part, this is actually the head part, and this is your tail part, and we have head meromyosin and LMM HMM அப்படின்றது வந்து light meromyosin and heavy meromyosin. Dystrophin protein complex connects muscle fibers. Yes, we'll discuss about the dystrophin very shortly. Mechanism வரும்போது we'll discuss about the dystrophin. Okay. Protein provides instructions for making a protein called, okay. Dystrophin ஓட இன்னொரு feature வந்து the molecular weight வச்சு ஒரு முக்கியமான ஒரு information இருக்கு dystrophin, low molecular weight, high molecular weight, அது என்னன்றது can you tell me? So that in that angle I'm asking dystrophin. Okay, that the function is correct. It provides instructions for making a protein called as dystrophin. That is correct. I'm talking on behalf of molecular weight. நீங்க இந்த ஹியூமன் ஜீனோம் ப்ராஜெக்ட் இந்த டைம்ல எல்லாம் படிச்சிருக்கும் போது புக்லயே கொடுத்திருப்பாங்க, டிஸ்ட்ரோபின் பத்தி படிச்சிருப்பாங்க. அப்போ அதுல கொடுத்திருப்பாங்க டிஸ்ட்ரோபின் பத்தி. இதுல ஒரு முக்கியமான ஒரு சென்டென்ஸ் வந்திருக்கும், 12th ஸ்டாண்டர்ட் புக்ல இருக்கும். ஸோ டிஸ்ட்ரோபின் பேஸ்ட் ஆன் இட்ஸ் லைக் மாலிகுலர் வெயிட் ஐம் ஆஸ்கிங். ஓகே, I'm not talking about the function, I'm talking related with its molecular weight. Other the shortest protein, low molecular weight protein, high molecular weight protein, are related with that I'm asking. Okay, so coming with the head part and the tail part. So HMM and LMM, that is light meromycin. There are two fragments that you'll get once you treat the myosin with the trypsin. This head consists of two sites actually. One is actually ATP binding site. This is the ATP binding site. The other one, this is actually, we call, what is other region like? So one is ATP. This is actually actin binding site. This is actin binding site. Fine. So LMM, HMM, light meromycin and heavy meromycin. So then also LM myosin வந்து two types இருக்கு, light meromycin, heavy meromycin. இது எப்படி actually double fragment, dual fragments actually. இந்த ரெண்டு filament மாதிரி இருக்கும். இந்த ரெண்டு filament பிரியும். எப்போ பிரியும்னு கேட்டீங்கன்னா when you treat with the trypsin. When you treat this myosin with the trypsin, this enzyme, this get the two fragments will get separated. ஒன்னு வந்து light meromycin ஆவும் பிரியும், இன்னொன்னு வந்து heavy meromycin. அதுல light meromycin வந்து functional இந்த மாதிரி binding sites இருக்காது. heavy meromycin ல மட்டும் தான் you could see the binding sites. Based on that only you can separate LMM and LMM and HMMM. Okay, so largest human gene, yes exactly. So DMD is actually the largest human gene. It's a பெரிய gene. Can you tell me like how many genes present in the dystrophin gene? Because often they ask this question, molecular biology போகும்போது dystrophinல எத்தனை genes இருக்குன்னு questions வரும். So how many genes which present? You said it's the largest known human gene. How many? Actually, what is the molecular weight and how many genes are exactly present in the dystrophin? So that is actually one of the important area of the dystrophin gene. For that only I asked this question. So this is actually like an actin binding site, and this one is actually your ATP binding site actually. So coming to this, like the size of the myosin is 150 angstrom. So ஒரு sarcomereோட size என்ன சொன்னோம்? What is actually the size of the sarcomere? The same way, the size of this myosin is actually 150 angstrom. The same way when they ask about the size of the actin is 70. Actin வரும்போது 70 angstrom units. But when it comes to the myosin, it is 150 angstrom units actually. Okay, so this consists of two parts. This is actually the head part. It consists of ATP binding site and the actin binding site. So next we move on to the actin. Actin. So the myosin is actually, this is for what type of band actually? Myosin. This is for A band. Actin is for what? Actin is a light band. This is a I band actually. This is for, this contributes like I band la அதிகமா இருக்கிற proteins எதுன்னு பாத்தீங்கன்னா actin இருக்கும். அதே மாதிரி மயோசின்ல வந்து டார்க் பேண்ட்ன்றோம், அதுல அதிகமா இருக்கிற பேண்ட், அது மெயின் A band அதிகமா இருக்கிற proteins என்னன்னு பாத்தீங்கன்னா it is actually the myosin. Coming to the actin. So when you look at the actin structure, so I'll just show the, instead of drawing in, see, this is actually the structure of like an actin filament. I hope it is visible for you. Fine. So looking at the actin filament, this like the globules, like this part, like spherical part is that you could see, right? This is actually, I'm talking about like the actin part. This I call it as a G actin. G actin, it gets converted into fibrous. Actually G in the then getting a globular. This globular actin which gets converted to this type of structure, this is called as a fibrous protein, and in the presence of magnesium ions. In the presence of magnesium ions, what happens, like this G actin, initially G actin or structure எப்படி இருக்கு, வெறும் globular ஸ்ட்ரக்சர்ல இருக்கும். இந்த G actin என்ன பண்ணுனா in the presence of magnesium this will form the helical like structures. அப்படியே வந்து the DNA ல இருக்க the helical structures மாதிரி என்ன ஆகும்னா it will be get twisted actually. So now this twisted helical structure is actually we call it as a fibrous protein. Initially it was in the form of what? It was in the form of a G actin. So along with the G actin we have like other two regulatory protein. Other two regulatory protein are troponin and tropomyosin. We have other two protein, that is troponin and tropomyosin. What is the significance of G actin? G actin which consists of what is actually the myosin binding sites, which consists of myosin binding sites. ஒரு மசில் வந்து கான்ட்ராக்ட் ஆக போகுது அப்படின்னு கேட்டீங்கன்னா எப்படி கான்ட்ராக்ட் ஆகணும்னா there should be a coordination between your actin and myosin. ஒரு பாயிண்ட் ஆஃப் டைம்ல உங்க ஆக்டினும் மயோசின் என்ன ஆகணும்னு கேட்டீங்கன்னா it should join actually should contraction ஆகும்போது ஆக்டினும் மயோசினும் மீட் பண்ணணும். அப்போ ஆக்டினும் மயோசினும் வந்து எந்த இடத்துல மீட் பண்ணணும்னு கேட்டீங்கன்னா இந்த ஆக்டின் that is like in this region there is a specialized part is called as G actin part. இந்த G actin partல தான் there is a particular place where you could see the myosin head. The previous headல நம்ம உங்களுக்கு சொன்னது பாத்தீங்கன்னா மயோசின்ல வந்து இந்த ரெண்டு ஹெட்ல வந்து ஒன்னு வந்து we call it as ATP binding site, the other one is actually we call it as a this is actually actin binding site. இந்த actin binding siteம் இந்த G actin இருக்கு பாருங்க ரெண்டும் வந்து ஒரு point of timeல this will come and bind actually. So that is the place where the contraction takes place. When the binding the power pack form then the ATP will come and bind. இது வந்து separate வந்து we going to discuss under the sliding filament theory. Next class like maybe tomorrow we'll discuss about the sliding filament theory. There you will clearly understand. நீங்க அதை அண்டர்ஸ்டாண்ட் பண்ணிக்கணும் அப்படின்னு கேட்டீங்கன்னா now you should clear with the structure actually. மயோசினோட ஸ்ட்ரக்சர் வந்து ஹெட் இருக்கு டெய்ல் இருக்கு. ஹெட்ல வந்து ரெண்டு பைண்டிங் சைட் இருக்கு. what are the two binding site? ATP binding site and the actin binding site. அதே மாதிரி மயோசின் வந்து எந்த ரீஜன்ல போய் எந்த ஆக்டின் ரீஜன்ல போய் பைண்ட் ஆகும் அப்படின்னு கேட்டீங்கன்னா there's a specific site called as G actin. So you could see this is actually this complete site is actually we call it as what? It is a actin. This complete site is called as actin. இது வந்து complete வந்து actin. இது வந்து complete F actin, that is like fibrous னு சொல்றோம். இதுல ஒரு particular region இருக்கும். This particular region where you could see beneath இதுக்கு இங்க இருக்கு பாருங்க இந்த இதுக்கு கீழ ஒரு particular area இருக்கும். இதுக்கு கீழ தான் என்ன இருக்குன்னா you could see something called as G actin. இந்த G actin யாரு கவர் பண்ணிருப்பா அப்படின்னு கேட்டீங்கன்னா you could see this thing actually. இங்க இருக்கு பாருங்க this globular structures you could see the troponin. இந்த troponin என்ன பண்ணிருக்கோம்னா G actin கவர் பண்ணி வச்சிருக்கோம். எப்போ ரிலாக்ஸ் கண்டிஷன்ல இருக்கிற வரைக்கும். நார்மலா நீங்க படுத்துட்டு இருக்கீங்க எந்த ஒர்க்கும் பண்ணல அப்படின்னா முதல்ல என்ன ஆகும்னு கேட்டீங்கன்னா G actin வந்து கவர் ஆயிருக்கும். யார் அதை கவர் பண்ணிருப்பான்னு கேட்டீங்கன்னா troponin வந்து கவர் பண்ணும், மேல மாஸ்க் பண்ணிருக்கும். In case like you want to do any work அப்ப என்ன ஆகும் அப்படின்னு கேட்டீங்கன்னா the troponin will be unmasked actually. இந்த G actin வந்து troponin நகர்ந்துரும் பிறகு அப்ப என்ன ஆகும்? The G actin will be exposed to what? Exposed to myosin. Then what will happen? The myosin will come and bind onto the actin. Then only what will happen? The sliding filament, the contraction takes place. So normal relaxed conditionல இருக்கும்போது G actin அதாவது actinல இருக்கிற the G actin the region வந்து will not be exposed. ஏன் இது expose ஆக மாட்டேங்குது அப்படின்னா something is covering, something is masking. Who is covering? Is actually the troponin part. The troponin is covering during relaxed condition. Once the muscle is going for the contraction, this troponin like which will be moving out of that particular region, then what will happen? The G actin region will be exposed, then that particular part where the myosin head will come and bind, so the contraction takes place. இதுதான் நம்ம நாளைக்கு நம்ம என்ன பார்க்க போறோம்? We're going to discuss in detail about the sliding filament theory. I think like already time is 4:45. So time is up. So what we're going to do is like நாளைக்கு நம்ம டிஸ்கஸ் பண்ண போறது வந்து we discuss about the sliding filament theory. இன்னும் troponin பார்க்கல, tropomyosin பார்க்கல. A little bit of information. There are 3 types of tropomyosin இருக்கு. So அந்த tropomyosin ஓட டைப் பார்க்க போறோம். Then we'll discuss about like sliding filament, that is Anselin and Huxley அந்த டைப் பார்க்க போறோம். Then regulation, how like contraction relaxation. Once இது ஒரு 15-30 minutes முடிஞ்ச பிறகு then tomorrow we'll start with the skeletal system. ஒரு சின்ன ஹோம்வொர்க் என்னன்னு கேட்டீங்கன்னா skeletal systemல இருக்க because textbooksல 206 bones உங்களுக்கு கொடுத்திருக்க மாட்டாங்க. But I don't want to study all the bones. I just want to categorize actually 206 bones la appendicular skeleton and we have like an axial skeleton. Axial skeleton la skull எத்தனை bones இருக்கு, appendicular எத்தனை இருக்கு, axial skeleton எத்தனை bones இருக்கு. Just அந்த number of bones மட்டும் by some source you try to calculate, so it will be helpful for you. But for example நீங்க skull எடுத்துட்டீங்க அப்படின்னு கேட்டீங்கன்னா tomorrow we'll be discussing about this. For example, if you're talking about a skull, getting na. So if you talk about what are the bones, this part is actually called as axial skeleton. This is like appendicular skeleton. So what are the name of the bones that we're going to see, and what are the name of the bones, skull and a bones. So for example, 8 cranial bones. You must be knowing the numbers actually. If you just go through by today, like tomorrow, it will be helpful, so you can understand much better. Okay. So I think the time is up for today. So tomorrow we'll meet in bio class and we'll start continue with the muscular system, and also we try to complete this chapter along with the skeletal muscles. If any doubts or any queries, like you can ask me without any hesitation. So thank you for joining. Thank you so much.
சார் எண்டு ஸ்ட்ரீம் கொடுத்துடலாமா சார்? எண்டு ஸ்ட்ரீம் கொடுத்துடலாம்.